cell signaling is really important for all of the cells to communicate with each other. And then we need to study the pathways by which cell communicates, because it's necessary for all of the cells to come into place with each other. So we will be talking about different kind of pathways that cell use to communicate. Simple. So the major receptors, or the receptors used by different ligands, we're going to study those, and then I will be elaborating those. So receptors that we'll be studying over are GPCRs, which are G protein coupled receptor. We have receptor tyrosine kinase, and then we also have tumor necrosis factor. There are other, but like ligand, gated channels, and then steroids, which affect nucleus. But we are not going to be talking about that. We're just going to focus on these three right now. So if we come right here, we have a GPCR. G protein coupled receptors is usually about, it's a protein molecule that is embedded in the cellular membrane, as we can see right here. It usually consists of about 7 helices going in and out of the cell, and it has a receptor site, and it has like a, you can say it activates other molecules which are also called second messengers. So what happens is you have your ligand. So for example, let's consider this ligand. It's going to come in and bind it. As soon as it binds it, this thing is going to activate a protein molecule known as, it has 3 subunits. So it got alpha, beta, and gamma. So alpha subunit is a GDP or GTP binding domain. So it binds GDP or GTP. So as soon as the ligand binds to the GPCR protein, it causes this GTP to be replaced by GTP, causing them to dissociate. So when they dissociate, they become activated. So now this is activated and this is activated, and they can go further downstream signaling to cause changes in the cell in different pathways. And we're going to So now we're going to discuss about glucagon. What is glucagon? Glucagon is a hormone released when glucose is gone. Well, you got glucagon, which is a hormone. It acts as a first messenger, binds the GPCR protein. It acts via GPCR signaling pathway. It comes and activates GPCR, which causes this subunit, like alpha, beta, and gamma subunit, to dissociate and get activated by binding of GTP. When it binds GTP, it's known as GS alpha, which means it's a stimulating behavior. It has a stimulating behavior. This causes or recruits a molecule called adenylate cyclase, which is a very common molecule throughout the cell because it plays a really important role. It converts our energy, which is ADP, which is $. It moves ADP and changes to CAMP, which is CAMP. This molecule activates another molecule called protein kinase A, which causes a further signaling that increases glucose export. So you see how one thing, which is GPCR, can make multiple changes downstream signaling through second messenger molecule to increase something which should have been increased because there's less glucose in the blood level. So it increased glucose like this. So now you'll be wondering, how does this whole process end? Now it's a really good question. What happened is this GS alpha, which is G protein stimulating alpha subunit, is a GTP binding and is activated. So what happened is you have this scary looking dude known as GAPS, or GTPase activity. It has a GTPase activity, meaning this can snatch this GTP as it is money. It can snatch it away from this alpha protein, causing it to deactivate it by binding the GDP instead of GTP. So which releases a phosphate group, deactivating this whole thing. So now we're going to talk about another kind of receptor, and it's a pathway signaling just like GPCR, called receptor tyrosine kinase. So in this case, they're usually monomers. So they're right here, they're also in the cell membrane, they're monomers. So when the ligand binds to them, they find another monomer and dimerize, meaning that they find another one and they stick together. And then they do one thing really special called autophosphorylation. So one monomer cross-phosphorylate the other one, and the other one does the same thing. So you scratch my back, I scratch yours. And then now they have these phosphate groups on them. So these phosphate groups activates the whole dimerization molecule receptor tyrosine. And then they recruit a bunch of buddies. Those buddies include an adapter protein, usually SH3, SH2, which adapts another protein called son of seminless. That son of seminless, or also known as GEF protein, activates, actually it recruits RAS and activates it by making it go from GDP to GTP. And that GTP is brought by the same protein that we were talking about, and and it kind of catalyzes it to have GTP, causing it to be activated. And then it can go through further downstream signaling to produce other effects in the cell, which usually we're going to talk about next is MAP kinase pathways. These are a bunch of molecules which phosphorylate each other to get to ERK, phosphorylated ERK, to gene regulation. So they are the one which can go into the nucleus and regulate gene regulation. So they can regulate how genes are expressed or repressed. So now we're going to talk about the example of tyrosine kinase receptor, or RTK in short. And the major hormone which uses this pathway is insulin. So insulin, when binds to the receptor tyrosine kinase, it does the same thing. It dimerizes, or if it is already dimerized, then it autophosphorylates it, and you got these phosphate groups. Then it recruits its buddies, which include IRS, which is insulin receptor substrate. That is a adapter protein, which then recruits or phosphorylates PI3K, which then convert PIP2, which is phosphonylnosyl phosphate 2, to PIP3. So having from 2-phosphate to 3-phosphate, and the enzyme that is being used is P10, which is known as phosphate and tensin homolog. That converts PIP2 to PIP3. Now PIP3 plays an important role. It also helps PDK1, which is phosphodiinositide kinase 1, to phosphorylate AKT. Now this AKT can go in and stop glucose from excreting and increase the glucose intake from the blood into the cell, reducing the glucose in the cell. That's why insulin is really important, and for diabetic patients especially. I promise this is the last one. This is the last one, and known as tumor necrosis factor. It is really important in regulating the cell survival versus cell death. It causes what we call programmed cell death, and that means that this uses certain kind of proteins to kill the cell. It is very important for cell to regulate the cell death versus cell survival. Any imbalance in between these two can cause or lead up to tumor growth and eventually cancer. These also works in the same way. You got a receptor which is bound to the membrane. There's usually ligand or ubiquitin which binds to them, and then they recruit. These are called death domains right here. These are called death domains, which recruits TRAD and FAD. TRAD and FAD makes a complex, and that complex actually recruits procaspases 8. This procaspases has a group attached to it which is cleaved to make caspases 8. This caspase 8 cleaves another group on another procaspase known as procaspase 3, and it cleaves that group from right here and making it caspase 3. Now caspase 3 is actually our killer. It goes to the cell and literally terminates it. It causes cell death. Now there's another mechanism for this where it can regulate this by inducing cell survival. But the major part of tumor necrosis factor is to regulate this for cell signaling pathway to kill or to survive the cell.